of aging usually centers on two specific anxieties: the gradual loss of our mental sharpness and the physical decline of a crumbling body, to wear out. This belief drives us to seek out lives of maximum comfort. We sit in ergonomic chairs and optimize our climate control, attempting to shield our bodies from any unnecessary strain or damage. But if we have fundamentally misunderstood why our bodies age, then these efforts to protect ourselves might be having the opposite effect. Take the moon jellyfish. These organisms possess a degree of biological immortality. They can actually revert their cells back to an earlier stage of development when threatened or injured. If some species can bypass aging altogether, it suggests the process isn't a universal law of physics, but a biological process that is managed within our own cells. Without the pressure of external stress to trigger repair, a sedentary life signals to the body that maintenance isn't a priority. Consequently, our comfortable routines allow cellular trash and DNA damage to accumulate without a system in place to clear them out. By treating our bodies like fragile machines that must be shielded from the world, we are likely accelerating our own decline. To understand how this happens, we have to look at the epigenome. This is a layer of instructions that sits on top of our DNA, telling each individual cell which genes to read and which identity to take on. At the cellular level, aging is essentially a loss of information. As the epigenome flickers and degrades over time, our cells begin to forget their identity, leading to the malfunctions we recognize as aging. We can actually measure this decline by tracking telomeres, the protective caps at the end of our DNA strands that shorten as our biological clock ticks down. Because the underlying DNA often remains intact while the instructions fade, it suggests that aging isn't an inevitable hardware breakdown, but a loss of cellular data. In 2006, Shinya Yamanaka identified specific proteins capable of totally reprogramming mature cells. These Yamanaka factors eventually earned him a Nobel Prize and opened a new door in longevity research. Recently, researchers applied these factors to an old mouse suffering from age-related blindness to see if they could influence the epigenome of a living mammal. The experiment worked. The Yamanaka factors successfully reset the epigenome in the mouse's eye, forcing the cells to remember their original identity and entirely restoring the animal's vision. The fact that these cells could be prompted to return to a youthful state suggests that aging may not be a one-way street. While gene therapy of this kind isn't yet available for humans, we already possess a built-in repair network, a family of longevity genes called sirtuins. The problem is that our modern, climate-controlled lives keep our bodies in growth mode, where cells focus on dividing and copying data rather than pausing to fix existing errors. To wake up these sirtuins and trigger the repair process, we have to move the body out of its comfort zone using deliberate biological stress. This intentional challenge is what forces our cells to stop growing and shift into their innate maintenance mode. You can trigger this reboot through thermal stress. Practices like intense cold exposure or extreme heat from a sauna provide the sudden shock necessary to signal the repair network. We can also achieve this through caloric restriction. By simulating scarcity through fasting, we provide another powerful signal for the body to prioritize its own survival. These stressors prompt our cells to clear out cellular trash, repair DNA damage, and begin restoring the lost information in the epigenome. Aging is no longer a process we simply have to endure. By mastering biological stress, we have a tool to potentially hack, reset, and influence our own biological clock.